Hi, my name is Anna Rodriguez, and today we decided to do our presentation over the paper Capsaicin-Shaped Gut Microbiotic and Pre-Metastatic Niche to Facilitate Cancer Metastasis to the Liver. To give some background information, diet plays a critical role in the development of gastrointestinal diseases and cancers. This is because the gut microbiome, which is the bacteria within our intestines, influence the immune system and liver health. Spicy foods, whose main component is capsaicin, has been linked to both positive and negative health effects. Many studies have shown that capsaicin has tumor suppressant functions as inhibiting cancer cell growth and reducing tumor development. However, other studies such as this one suggest that capsaicin can promote tumor genesis. Cancer cells don't randomly migrate to distant organs. They tend to go to places where the pre-metastatic niche has been formed, and a pre-metastatic niche is an area of the body that has been primed to support incoming cancer cells through inflammation, immune suppression, and microenvironmental changes.
Hi, I'm Ella, and I'm going to be doing the introduction and figure 1. So there's a few main points for the introduction that I was going to cover. First of all, capsaicin is the main active ingredient in a spicy diet and is being analyzed in this overall research for its role in promoting gastrointestinal diseases and colon cancer. So previous studies have shown that capsaicin has tumor suppressive functions, which makes this a little more complicated because it's hard to kind of disprove other research. So this overall asks the question of whether low dose or high dose of capsaicin in the diet influences cancer metastasis, and it also uses the correlation of the microbial compositional changes in the intestines and the immune microenvironment in the liver, which are affected with intestinal function. So overall, the main goal of the research and the main conclusion was that long-term administration of capsaicin disturbs the composition of gut bacteria, the liver bacteria, the gut vascular barrier, and bile acid metabolism.
Hi, Mariana, and I will be doing the methods. Lab mice were fed either a control or capsaicin-enriched diet for 12 weeks, then used in a liver metastasis model with EGFP-labeled CT26 cells. Researchers assessed tumor burden, gut barrier function, and bacterial translocation using imaging, histology, and molecular assays. Microbiota composition and bile acid profiles were analyzed through sequencing and mass spectrometry, while flow cytometry and fecal microbiota transplantation evaluated NKT cell recruitment and the role of gut microbiomes in metastasis.
The original hypothesis presented for this figure was the pro-inflammatory nature of the high-dose capsaicin would form a pre-metastatic niche to support cancer cell metastasis. In order to make a conclusion for this hypothesis, a capsaicin diet was introduced to mice before injection of tumor cells for 12 weeks. The result was higher inflammatory cytokines, as shown in parts B through E, and upregulation was also seen in expression of transforming growth factor beta and other liver components. Overall, this shows that the inflammatory microenvironment formed after long-term capsaicin intake. The diet also increased liver infiltration of immune cells such as neutrophils, macrophages, and monocytes, with NKT cells being reduced in the liver. Part I through K also shows there was a huge increase in liver tumor nodules and more metastatic components as seen through the bioluminescence. That is more towards the eye, and it shows different parts of the liver. So in summary, this figure shows that high-dose capsaicin could promote colorectal cell metastasis to the liver through the inducing of a pro-inflammatory metastatic niche.
In this figure, we are being demonstrated how capsaicin is disrupting the intestinal barrier and promotes bacterial translocation into deeper gut layers. In figure A and B, it shows H&E stained tissue sections from the colon, the cecum, and the ileum taken from mice after 12 weeks of a capsaicin diet, with each mouse taking different amounts. Within this figure, we can observe chronic inflammation and tissue injury within each tissue section. Figure B is used to quantify the damage done to the tissue section. It resulted in the mouse having a higher intake of capsaicin to have the most inflammation and injury to its tissues. In figure C and D, they are using ONC and blue staining to show the goblet cells within each tissue section. In figure C, we're able to see less blue staining within the tissue as the intake of capsaicin increases, which overall suggests that the high capsaicin intake causes a reduction of thickness within the mucus layer. And in figure D, it's just using to quantify those results. In figure E, they're using immunofluorescence analysis to stain for the expression of tight junction protein ZO1, claudin 1, and cell adhesion proteins E. coherin. Within the image, we're able to see the colors be less visible as the intake of capsaicin increases. Due to the decrease of these proteins, we're able to conclude that the barrier is breaking down. And in figure F, FITC dextran, a fluorescent molecule that can pass through healthy gut lining, was used to determine the gut permeability. They were able to find that as the intake of capsaicin increases, the more of the molecule was found within the serum, indicating that there is a disruption within the lining and the molecule has become more permeable. Lastly, in figure G and H, bacteria was stained by fluorescent in pseudo-hybridization to detect the presence of bacteria. The results that the bacteria has translocated from the intestinal lumen and penetrated into the submucosal layer. In figure H, it is used to quantify those results, showing that bacteria has translocated in the mouse with the highest capsaicin diet intake.
In figure 3, the researchers looked at how bacteria in the gut were able to cross both the intestinal and gut vascular barrier, or GBV, which is a layer of blood vessels that normally prevents bacteria from entering the bloodstream. They found that increased levels of PB1, a protein that marks GBV distribution, and saw more PV1 plus CD31 plus endothelial cells, indicating vascular damage. Using fluorescent and C2 hybridization, or FISH, they actually visualized bacteria in the liver, mostly outside immune cells, and confirmed their presence through colony-forming unit, or CFU, assays, meaning viable bacteria had migrated from the gut to the liver. In figure 4, the gut microbiota changed significantly at capsaicin-fed mice. 16S rRNA gene sequencing revealed a reduction in Akkermansia, a genus known for supporting gut barrier function, and an increase in Mirabaculaceae, which can degrade the mucus barrier. This imbalance likely contributed to the damaged gut lining. There were also changes in other genera like Rumini-Clostridium and Bifidobacterium that are involved in bile acid metabolism, hinting at a deeper impact on the liver's immune environments.
Hello, my name is Yurim Cho, and I'll be talking about Figure 5, which this figure shows how capsaicin alters bile acid profiles via the microbiota. Figure A to C uses the LC-MS bile acid profiling methods, which show that capsaicin increased the THDCA, GHDCA, GUDCA, which decreased the alpha MCA and the DHLCA. Figure D uses the bile salt activity assay, which shows the BSH activity increased with capsaicin dose. Figure E shows the fecal bile acid quantification method, which shows that more total bile acids and feces of capsaicin-fed mice. And figure F shows the method using the quantification PCR of microbiota, which shows more bile acid converting bacteria with capsaicin. Figure G also shows the qRT-PCR of liver mRNA, which shows the CXCL16 gene that's upregulated by THDCA and GUDCA, and others that are suppressed by the alpha MCA. Then we're trying to see how bile acid receptor activates reverses effects. Figure H shows the rescue experiment with OCA treatment, which results in OCA administered after capsaicin exposure. Figure I to K uses flow symmetry, which results in OCA reverse capsaicin-induced immune cell recruitment to liver. And figure L also shows or uses flow cytometry, and you can see that there's no significant change in macrophage numbers. And lastly, figure M uses the tumor counting method, which results in the OCA reduced liver metastasis in capsaicin-treated mice. Next, we will move on to figure 6, this experiment is generally done to see if capsaicin alters gut microbiota and increases gut permeability. For figure 8, the fecal microbiota transplantation method was used, and in the results, we see that the CAP control mice received microbiota from capsaicin-fed donors. And figure B then uses the immunofluorescence staining method, which was used, of which the capsaicin-controlled mice had disrupted tight junctions in ileum, which means that there was a weaker straining. Then in figure C, it was using the FITC dextrin permeability assay, which shows that the capsaicin-controlled mice had a higher serum FD4, which means that it has a leakier gut barrier. Now they're trying to see how the capsaicin microbiota promotes liver inflammation. Figure D to F uses ELISA method of the liver tissue, which shows an increased TNF alpha 2,6 and IFN gamma in capsaicin-controlled mice. Figure G shows flow cytometry, which showed more NKT cells in capsaicin-controlled livers, showing immune activation. Then they're trying to see if capsaicin-altered microbiota enhances liver metastasis. As you can see in figure H, we use the cancer model CT25. GFP cells injected post-FMT. This results shows the CAP-CAP mice used to test pro-metastatic niche. Then figure I uses gross imaging and bioluminescence, which shows that the CAP-CAP mice has more liver tumors and stronger GFP signal. Lastly, figures J to K use tumor count and radiance quantification, which shows that the CAP-CAP mice had significantly more nodules and tumor broaden. Lastly, I will conclude this whole project, which firstly we found that long-term high-dose capsaicin intake promotes liver metastasis of colorectal cancer by modulating the gut microbiota and disrupting the gut-liver axis, and it also altered the gut microbiota, and the capsaicin also damages the intestinal and vascular barriers, and there were skewed bile acid metabolism, and lastly, it creates a pre-metastatic niche. Thank you for listening, and I will now take questions.